Well, thank you so much for inviting me, and I hope that you're enjoying your natural reward of food now to feel relaxed after lunch. My College of Pharmacy colleagues told me that if you can't understand my accent or any difficulties, I'm from the north of England. One claim to fame is that I went to high school in the same town where Daniel Craig, the latest James Bond, went to. But anyway, we speak basically the same language, so hopefully you'll enjoy this talk today. I will say, though, I probably don't have all the answers today. We're really living in a very rapidly changing environment in terms of how we're going to deal with marijuana. But my hope is to explain some of the pharmacology, because as a basic scientist, I think that there is great hope that we will eventually use the mixture of natural products that are in marijuana to perhaps inspire new drugs, and that we may move on and actually have some clearer ideas with respect to how we may manipulate this signaling pathway for the treatment of things such as chronic pain. So, just as an outline to my presentation, and we will talk about the pharmacology of the cannabinoids. We'll talk about some of the recent trends in marijuana use, and this again is also just like similar principles to other substances that are abused. There are always new things coming out, so just trying to keep in touch with latest trends. You know, it probably will be different 5 years from now, but as far as I can give you a brief overview on how it is being used recreationally and by the pain medically as well. We'll look at the approved uses and the rationale for the use, but here I'm going to stick specifically in pain, because we've got about 10 approved uses in terms of the debilitating conditions that are recognized by the state of Oregon, but I'll only focus on pain today, just in the interest of time. And then towards the end, we'll just talk about the potential for drug-induced short and long-term effects. So this really should really fit in nicely with some of the information that Dr. Hedberg talked about earlier this morning, but just to expand a little bit on that so that you all feel that you're familiar with the long history that we have in Oregon for marijuana. So in 1998, the Oregon voters approved ballot measure 67, and that seems a long time ago now, but really what that did was allow the use of marijuana for debilitating medical conditions, and established, sorry, I don't think my pointer's working very well, established a state-controlled permit system to administer and oversee this act. And that is still under or overseen by the Oregon Health Authority. And so actually the Oregon Health Authority website is very useful as a source of information that, as you probably learned from Dr. Hedberg's talk, they're continuously updating the guidelines. So as a resource, that's very, very valuable for all healthcare providers in the state of Oregon. So really the idea of overseeing this, what became or is known as the Oregon Medical Marijuana Program. So we have an act that allows Oregonians with these conditions to discuss the use of marijuana, and also the idea is to encourage that conversation, protect physicians and the patients themselves from penalties, because as we all know, at the federal level, this is still a controlled substance. And so we've yet to see how this pans out under the new administration, but right now we have the act in place. So very recently, relatively recently, when you think of the history of the College of Pharmacy in our state, where we the normal track for dispensing prescribed medications, we have actually got a whole new system that has grown up, and as of January 2014, the Oregon State Legislature approved the establishment of the medical marijuana dispensaries, also that are overseen by the Oregon Health Authority. So this number is down a little bit at the moment. Sorry, I'm I'm ignoring everyone on this side of the room. So down a little bit to, the statistics are up there now. As of January 2017, we've got about 307 dispensaries and 121 processing sites that are registered with the Oregon Health Authority. Now July 2015, it's now not last year, recreational marijuana became legal for personal use in Oregon. In October, then we had early sales of recreational marijuana at the medical facilities. And the idea was that this was now legalized in Oregon, and Oregon had a well-established dispensary system. They'd had a 18-month head start. These dispensaries exist in towns. We do get people driving from one town to the other, because there's some stigma perhaps in going to visit to your local dispensary. But this was to allow access for the recreational market or the retail market. Currently, in the dispensaries that sell both medical and recreational marijuana, that business end of it is kept quite separate, and as I'll mention in a little bit later, the medical patients have access to much greater amounts than the retail, and so the limits are set quite differently. So again, the idea is that the retail market for recreational marijuana will or is now under the control of the Oregon Liquor Control Commission. And I believe there's even a move to perhaps rename that in Oregon to the Oregon Liquor and Cannabis Commission, so we'll see what happens there. But then now the idea was that that early market, early access to retail marijuana was only temporary. Now to sell to the retail market, you have to have an establishment that is registered with OLCC. So I think that this slight dip, down from about 400 to 300 dispensaries is many dispensary owners that were only selling medical marijuana feel that they may not be able to make it and as a sole medical purveyor of medical marijuana. And so now this is all kind of a changing scene, and we've got a lot of medical dispensary owners now applying for their retail license. And so we've got this separation, Oregon Health Authority for the medical versus the OLCC for the retail market. So in trying to come up with a way to think about this, because and how we might realize where we are in terms of the research here and how we're going to make sense of it, I decided to look back in history and take a historical perspective. Because as you know, we have used natural products, terrestrial mostly, from the plant world, as inspiration for many of the pharmaceuticals that we use today, especially in the areas of cancer and infectious disease. But so it's quite interesting to look at this text that's in the National Library of Medicine, these beautiful pictures, and recognizing that this is an American medicinal plant, so cannabis grown in America, dated in about 1887. I guess I can look at it here as well. So beautiful old text. And I think historically we've never really been able to do a good job with these plants that have there are a subset of plants with medicinal properties which also have abuse liability. And that's really the main subject of today's conference is about this very famous one on the right, the opium poppy. But again, if we just look from 1817 to 1839, these all exist as historical texts, recognizing that there are chemical substances that do things to human behavior and or health. So we've got the tobacco plant. Obviously we've got nicotine addiction as a huge issue. We've got recognition of alcohol. Before anesthesia, we would drink a bottle of whiskey before somebody sawed your leg off. So, the idea of sedative hypnotics, or recognizing that these natural products, and I use the term natural products in that they are chemicals that have been that's already made in nature, and this has been our inspiration for many drugs. So I feel that probably we are in a similar situation with marijuana right now. We just don't really know. The research isn't there yet to tease apart exactly how we're going to use it, but I think there is probably some potential. And if you think about the idea of taking, this is what we do, we're really still doing this approach. If you think about how paclitaxel for some solid tumors has changed our treatment of some cancers, and recognizing that the bark of the Pacific yew contained a chemical substance, and that whole story of the development of paclitaxel is almost a little bit like, perhaps where we are with marijuana, but another good example. Really, if we took the mixture of compounds that are in the plant that produces cocaine, by the time somebody finally isolated cocaine and synthesized it, this really gave us our very first idea of a first local anesthetic. So that structure, even though finally we realized that as well as having abuse liability, it also binds to voltage-operated sodium channels. And then we got the development of procaine, lidocaine. This gives us our first natural product, local anesthetic. And I really love this poster from about 1885 for cocaine toothache drops available from your druggist, and I'm sure it really did cure your toothache. But if you think, though, of a historical perspective on how we can look to nature to understand and perhaps utilize for therapeutic advantage these kinds of chemicals. So even medicinal marijuana, and I'm probably using those terms a little bit interchangeably, because in Oregon we tend to use the word marijuana, although technically we should be talking about cannabis. So even American cannabis, so cannabis grown in this country has a long history of use. So back in 1854, it was listed in the US dispensary, and we had tinctures of cannabis. These would be very common things that physicians would carry in their leather cases, and would be part of the tools or medications that could be prescribed. They are also made in America by some very companies that we recognize today, Park Davis, Eli Lilly, Bristol Myers Squibb, Merck, and prescribed for such things as insomnia, headaches, sexual dysfunction of both sexes apparently, and eating disorders. So there's a hint as to some of the kind of classic things we know marijuana does in terms of stimulating appetite, perhaps. So then it tends to fall out of favor. So by 1937, the Marijuana Tax Act comes into place, with a lot of taxation on it and increased scrutiny, and then it is removed from the pharmacopoeia in 1941. So I think it's quite interesting. This is, I'll come back to this later in the talk. You said it does say poison there. The poison is because it's an 80% alcohol extract, I think. But we do have an old tincture of cannabis, and obviously was socially acceptable at that time. So really, what is medical marijuana? I think this is a good way to think about it. This is just really a very general term. So the cannabinoids are produced or found endogenously, and this is now the system that we refer to as the endocannabinoid system. And I'll spend some time talking about that, because that really gives us our greatest hint that there may be some true biochemical interactions that we can manipulate, especially in terms of pain. We can use the term, and especially in Oregon at the moment, when we say medicinal marijuana, we really mean these plant-derived natural products. Excuse me, from the phytochemicals that come from the cannabis plant. And some of our famous ones would be the delta-9 THC and cannabidiol, that we really are very focused on what these do in the body and what their targets are. And then perhaps I believe that these are prescribed much less in Oregon and much less these days, but we do have synthetic grade pharmaceutical forms of a delta-9 THC and a close isomer that have been around. They've been approved through the normal process. So really, those would be, medical marijuana is really an umbrella term for those three different kinds of cannabinoids. So why does the plant make these substances? Well, actually, that's very common. Lots of plants make a multiplicity of chemical compounds, and the marijuana plant has been greatly researched. We know exactly how many compounds are in that plant, and really this is just a natural biosynthetic pathway. So the plant is making this probably, or these cannabinoid compounds that we're interested in, as a secondary metabolite. So this is not life or death for the plant. This is allowing probably the plant to adapt to its environment. So in terms of the marijuana plants, the growers who grow in greenhouses near the Portland area will say their cannabis is very different to the ones who grow in southern Oregon, out in the open, where they don't need to irrigate, and just all the conditions, the irrigation, water, sunlight, growth period to me, it's no surprise that I believe Oregon is a great place to grow these plants, and we have lots of different kinds of plants because the plant will make different kinds of these components in different varying amounts. What seems to be gaining a lot of interest, certainly in the growers and the users, it's almost like the end user very often knows a lot more about this than we do as the healthcare professionals. And I think that's a dance that we want to kind of engage in those conversations so that we can learn from them. But it's almost in some places, some of the retail establishments and the medical marijuana establishments, sometimes it's likened almost to wine growing. You know, do you want this high concentration of terpenes? It's going to slightly change the way this product feels. And that does pose a big challenge for us. How do we know? What is a standardized product? As we're used to something being very regulated, produced in known quantities. We know what the dose is. And this feels, this vintage almost feels like talking about wines and menus. There's menus of this. To try and navigate this can be difficult. So it helps me just to realize that they're going to be very different. It may be very hard to standardize unless the plant is coming from the same place. And I guess this is what the National Institute of Drug Abuse was trying to do when they mandated that if you were an NIDA researcher, your strain had to be from the plant, that your product or what you're going to research comes from this one grow site in one state, and trying to regulate it that way. Whereas in Oregon, certainly the research done on those may not be applicable to what our Oregonians are using, because we've got a huge diversity there. The plant will be completely different. As I've learned with having the opportunity to even talk to some of the growers, they're very proud of what we have here and what we can do here, and how well we can manipulate and grow. You know, we're in an agricultural state, and so we've got a really diverse group of people that are feel very strongly about this topic. So I think I mentioned that we know there's not much new chemistry to explore, really. It's only the differences in the ratio of these plants. So we know that basically there's well over 400, probably at least, you see various estimations by different labs. So this is taking that whole mixture and asking how many different chemicals are in it. So it's definitely a polypharmacology situation. We've got lots of chemicals, probably at least 60 to 90. At least a quarter of them are what you consider the cannabinoid-like structure. But if shortly before it was removed from the pharmacopoeia, some of the main, more famous cannabinoids were isolated. So in the 1930s, cannabinol and cannabidiol. And then not till 1942 did people isolated delta-9 tetrahydrocannabinol, THC. So as often happens in natural products research, it took at least about 20 years for synthetic chemists to then be able to make that compound. So here's nature inspiring a structure, and then chemistry can go back and make it. So in the 60s, and then, just because the historical time frame I think is important, what this did was when it became listed as a Schedule I controlled substance, that really put a halt to much of the research. So we're really we're almost suffering from that now. So it was really, from that point onwards, mostly studied in the context of something that is abused or has abuse liability, rather than exploring any potential medicinal benefits. Again, really fairly new kids on the block with respect to the kinds of receptors that we know. So if you've got an endogenous signaling pathway, if there's an exogenous ligand, you're looking for the receptor target. It really took to the beginning of in the era of molecular biology, when we could clone and work out what these targets were. But this is, again, another 20 years past. So the CB1 receptor being cloned in 1990, and then the CB2, which is sufficiently different than CB1 to be classified as its own receptor, and I think that's worth remembering. So you get a lot of people just assuming that this is basically the same receptor. So they are geographically distributed in the body, if you like. They're in different places, but there's enough differences between CB1 and CB2 that it is considered a separate subclassification. So just before we leave, just so I don't completely forget about these, and so the point here is, and I think, you know, you all know that most of your day is spent thinking about things that have gone through the normal approval process. And so it is worth remembering that these do exist, and there was demonstrated efficacy. We've had these on the market. So there's 2 single molecule oral drugs. And so once there was a synthetic route to producing delta-9 THC and an analog, then it becomes economically viable to produce a single molecule. That single molecule now is interesting to think about that, because we've got many cases and some new therapies coming where we think it's not potentially just the single molecule, but this synergistic effect of more than one cannabinoid together. And that becomes difficult when we know there are so many. So the argument that the single molecule drug does not work as well as the mixture, I think, is believable if you think that there's probably a synergistic or combined effects of all those multiple chemical structures that are in the plant. Yeah, sorry. Yeah, so of course, instead of the normal approval process, this is our main problem, and which I think most of us in the healthcare profession currently have such difficulties with, because there are really no good, we're still in a paucity of information as to how, what kind of clinical data is out there where we can say this has been done in a proper controlled trial, randomized trial. We just don't have the benefit of these kinds of studies for this kind of product, which is very widely available. Even if you don't hold a medical card, we've got a lot of reduction in stigma. A lot of people now have access through the retail market and can try it out. And so I think we're really in a situation where people are using it, and finding out and asking those questions is probably more important than ever, because of the reduced stigma. But at the end of the day, we've got something that just hasn't gone through these normal approval processes. So if we think, just to think why is it people are drawn to particular strains? Just to think about what it's like as a patient or someone that is going to those dispensaries, and what it is they're purchasing or using. So really, delta-9 THC is what we consider the main psychoactive ingredient in this particular strain, cannabis sativa. So 2 of these main strains, cannabis sativa and then cannabis indica, are kind of recognized as kind of stock strains, but now we have a multiplicity of different hybrid strains. Indica was particularly favored for its very high THC content. So we've got multiple versions or growth, so they look quite different, but really both strains that produce the whole series of cannabinoids, including and favored for their high THC content. So THC as an exogenous ligand will bind to both CB1 and CB2 receptors with very respectable affinity. So if you look at the dissociation constant, so this has been measured by labs all over the world, and it turns out to be in the range of 5 to 18 nanomolar. So really a pretty good high affinity binding reaction when you take in this exogenous phytocannabinoid. So what CB1 and CB2 are G-protein coupled receptors. So they sit on the plasma membrane. And I think always the surprising thing is, because considering we only cloned this in 1990, we then can quite confidently say it's supposedly the most abundant G-protein coupled receptor in our brain. And that's really quite sobering when you think of all the pharmaceuticals, all the drugs that are out there that are manipulating other G protein coupled receptors, but just to think of it in terms of the abundance of this receptor, very, very, so that always will beg the question, well, what is it doing and how is it working? So CB1 and CB2 are the main class. We now, there's increasing evidence that there's probably orphan receptors. What we previously classified as orphan receptors, they're also G protein coupled receptors. But they are receptors that we know are also activated by exogenous cannabinoids. So at the moment, there's one G protein coupled receptor 55, which seems to be able to heterodimerize, or is also, there's homodimers and heterodimers with the classic cannabinoid receptors. It may in the future be reclassified as a CB3 receptor. We just don't quite know that yet. But it seems that the cannabinoids don't just act at these cannabinoid receptors. We know that now from trying to understand cannabidiol and how that works in the body. But anyway, these would be just classic G protein coupled receptors where the ligand binds to an extracellular domain and signaling is transduced across that plasma membrane to activate G proteins and produce a signal transduction event. So we know some of the very early studies. So about the time this was being cloned, so this is from Proceedings of the National Academy of Science back in 1990. This was the first really great study to localize and actually ask where CB1 is in the brain. And this is a little bit like those earlier heat maps we were looking of Oregon, only looking at imaging of the brain. So there were a lot of, the good thing, even though research into the medicinal properties of marijuana has lagged, a lot of imaging agents and a lot of ligands for these receptors, both agonists and antagonists, were being developed for a little while. There was great hope that CB1 antagonists may be useful as anti-obesity drugs. And so there was still a lot of those things being under development, and we got a lot of tools that we can still think of as valuable tools to look and image this receptor. So this is really the low, you can just see from this PET imaging study, which is taking a radioactive a tracer, in this case, to highlight. You can see there's an abundance of cannabinoid receptors in the brain. So the red to orange, a high density visualization of CB1 versus the blue areas. This first, if we look over, this is an antagonist, a tritiated antagonist in the days when autoradiography was a very kind of classic way to understand ligand receptor interactions. And so this data is normalized to the substantia nigra. And so we know that this cannabis use disorder and as something with addictive liability, we know that there's an abundance of these receptors in the substantia nigra, but these are looking at brain areas normalized to substantia nigra. And we can see that humans, just like the rat and the monkey, we have really cannabinoid receptors all over the brain, throughout the central nervous system. So I think one of the difficulties in studying it, or trying to, why we still are a little bit in the dark, we're still trying to work out how these things act. So why do we need so many cannabinoid receptors? What are they doing on our plasma membranes? Well, I think it's because we really have to think of them as neuromodulators. So unlike some of the transmitters that are responsible for very rapid signaling in our brains, such as glutamate or GABA, the GABA of these ligand-gated ion channels, or other G protein-coupled receptors, which act at kind of a really specific, they're released from a presynaptic side of a synapse, diffuse across, and have a real kind of strong receptor-target integration. These cannabinoids, or the endocannabinoids, really just modulate activity at the synapse. And so when you've got modulators, that automatically makes it more difficult to design assays to very accurately address exactly how they're working. And then if you think of the heterogeneity across brain regions, you've got we've got specific receptor targets, but place that in the whole human or the whole organism, and it's very difficult to actually understand what is going on. But I think we know now with quite good confidence that really the cannabinoid receptors are located presynaptically, and so that really helps in understanding how these work. So they're on presynaptic neurons to inhibit neurotransmitter release. So in theory, depending on which synapse they're on, they will have the potential to inhibit release of the neurotransmitter that is actually working at that synapse, the main neurotransmitter. So CB1 receptors are on neurons at the presynaptic site. The CB2 receptors tend to be primarily located on immune cells and throughout the periphery. So we do have this difference in the distribution of both receptor subtypes. I should mention, though, because sometimes this needs some clarification, it's not that CB2 receptors are lacking in the brain. CB2 receptors are on the microglia, which of course are a very important cell type that are performing an immune function role in the central nervous system. So it wouldn't be true to say that the brain is devoid of CB2. And so there's a lot of focus on the regulation or immune responses, and a lot of research being done on the role of CB2, particularly in microglia. So the brain is not this privileged CB1 site. It also contains CB2 receptor containing cells. So then a little bit later, we have the recognition. So if you find the target, what is the endogenous ligand for these receptors? And these were really the candidates that have been put forward shortly after the identification of the receptors with the endocannabinoid ligands. And we know now that they also may act on other targets. And so we've got a main system that we can study, but we acknowledge that there are probably a lot of off-target effects. So whether you're talking about the endogenous cannabinoids, and this slide is from actually Nature Reviews in Cancer, but really the principle is the same, whichever system we're talking about. So whether it's the exogenous plant-derived cannabinoids or the endogenous cannabinoids, the idea is that these are released almost as needed from membranes. So it's from lipid precursors. They're released into the synapse where they're able to interact with this presynaptic CB1 receptor. And that actually, through a sequence of events, decreases calcium and therefore decreases calcium-mediated neurotransmitter release. So that's really our working model at the moment for how we believe endocannabinoids or plant-derived cannabinoids will work. So, sorry, sometimes I'm not doing this quite right, I think. Try that way. Oh, sorry, I might need help. Try and go back. Sorry, I probably did something. Where's that? Did it go? Oh, yes, sorry. Oh, sorry. Sorry, I just it froze, I think. So just again because I think you always feel better if there's a true neurochemical mechanism of action behind these things, just to reinforce that what we think happens once the ligand binds to this receptor, we actually promote the opening of voltage-operated potassium channels located on that presynaptic plasma membrane. That inhibits the voltage-operated calcium channels, and then this is how we get presynaptic inhibition. So that presynaptic inhibition, again, this is, so again, even though we may feel a little bit upset that there's not progress in terms of clinical data, I think we are moving forward quite rapidly, really, in terms of understanding this at a ligand-receptor interaction. And so these are some of the endogenous ligands for the receptor. They're being released, and this is actually just taken from the ventral tegmental area. So especially looking at signaling in the reward centers of the brain and that ventral tegmental area to nucleus accumbens, that neuroanatomical region is really giving us a lot of clues. So whether you've got this cannabinoid receptor here, will potentially decrease glutamate release and glutamate effects at the level of the ventral tegmental area, or whether this be a GABAergic neuron that's interfacing with this ventral tegmental area neuron. At this synapse, this would potentially reduce the level of GABA. So we're really modulating major transmitters. So we think of glutamate and GABA as the major transmitters. These cannabinoids are modulating, just tweaking the environment really as needed. And I think the as needed will end up being quite important as we try to work through this and work out how they work. So the idea is that we now have two very kind of good candidates as anandamide and 2-arachidonyl glycerol, and they are derived from really synthesized as needed as membrane precursors. Most schematic diagrams at the moment seem to put these at the postsynaptic side of the synapse. So we've really got a reverse kind of signaling where we make the ligand as needed, and then that interacts with the presynaptic receptor to decrease neurotransmitter release. One area that's potentially very exciting in the future is that all the manufacture or the biosynthesis of the ligand requires enzymatic steps. So actually the formation of 2-arachidianylglycerol from diacylglycerol, that's quite an interesting area that when we know these enzymes and we know the degradation pathway, it turns out that we're going to end up seeing clinical candidates coming forward where we've got small molecules that can actually inhibit this degradation. So the idea, and we've used that strategy for a long time in terms of pharmaceutical development, where if you inhibit the breakdown of the endogenous transmitter, you would enhance functional levels of the endogenous transmitter at the synapse. And so this will be, in terms of a future druggable target, perhaps we might not always have to rely on ligands for the receptor. We can go after the enzymes that break down these particular ligands. So another exciting milestone last October, this was the first crystal structure has appeared in Cell, the crystal structure of the human cannabinoid receptor. So I think we've got a lot of very smart people working on this all over the world, and so hopefully this will help us if we're kind of working together. So this is the structural, no surprises, it's got predicted to have 7 membrane spanning domains, and then on the right-hand side, if we just look down on top of it, what this group, very huge collaboration of scientists from all over the world, they actually grew these crystals in a ligand and were able to report the first ligand-bound structure. The ligand they used was a very high affinity antagonist, and it did come from that pipeline of drugs that were being thought of for anti-obesity drugs. But the important thing is that they were able to see the crystal structure, see what the antagonist, this ligand-bound structure looks like, and from there start to predict with ligand docking experiments what the natural cannabinoids would look like. And so now there is the predicted models for what the delta-9 THC would look like, and what, as well as the phytocannabinoids as well as anandamide. The other interesting thing that I guess we've not mentioned it yet today, but from these structures, it's not really clear why things like K2 and spice, these synthetic cannabinoids that are coming onto, that are available, and we have guidelines and currently illegal, but it's not really clear why they have such a low therapeutic index, if you like. So why they are so lethal, whereas we know that delta-9 THC, when it binds to this receptor, it's not a lethal drug receptor interaction. So this has been very helpful and raised. Now we've got a working model, a template to move forward in understanding how to predict what would interact with this site. So in our state and in the states that have got marijuana available, people use this in a variety of ways. And I think we heard earlier in the day that we're never going to say that people should smoke this, but especially the older generation that don't perhaps want to try newer techniques, we still got, they're reluctant to give up smoking, or smoking of the hand, the kind of classic hand roll joint. Edibles, or smoking it, eating it, vaporizing, where we have inhalation of the vapor without some of the combustion products from the smoking, like we would a cigarette. Ingesting of edibles and teams. We've got topicals now, and these tend to be marketed for a local action right directly on the skin. So creams, lotions. It almost reminds me perhaps a little bit like Bengay, for acting on aches and pains. So we've got a topical market. Really what we mean by marijuana is really the dried flowers, leaves, and stems and seeds of that plant. So really a very complex mixture of products and many different ways to utilize it. So when you go into a dispensary, and again, your patients or users will probably know a lot more about this. I always, perhaps I'm a little bit naive, but at one outreach talk, somebody said, "Oh, it's okay. I know things you don't know, but you know things I don't know too." So there's always there actually is a nice to have a dialogue and learn from the people that know a lot about this. But most dispensaries contain or they have available many, many different strains, 50, 60 different kinds. But this now, we've got increasing scrutiny in terms of starting to require testing so that there's no contaminants, and making sure there's no residues of pesticides, but also the most, I hate to say users or patients are looking, they want to see a potency profile. And when the cannabis that's available for sale will have information that's associated with it. And normally there would be a % weight of cannabinoids versus the total plant weight. So this sort of boils down to some numbers, so it looks very different than we're used to milligrams in terms, and that would be the way to communicate dose in edible formulations. But here we've got percentages, and so really, anything that's greater than 14%, especially using especially if we think back to the 90s, this is changing as well. But really, the idea was that 14%, so that we don't kind of lose sight, or we may have to recalibrate ourselves, for what's being used out there right now. But really, anything above 14% was considered high THC. We've got some patients that are looking for much lower THC and instead wanting high CBD. So this is where some of the genetic and the agriculture is coming into it in terms of producing hybrid strains. So low CBD would be less than 1%, whereas high is around 4%. It's interesting to note that this is from the New England Journal of Medicine, a couple of years ago, but we already have, or we realize that marijuana, what is out there and is available for consumption, is growing in potency. So the market is that some people want high THC. We've got the agricultural means to do that and the knowledge of how to grow and make strains with very high THC. So this has been put forward as a reason for this growing trend in emergency room visits with respect to marijuana. So people tend to seek help, perhaps the experience or they're fearful of the reaction, or it was much stronger than they anticipated. And then this old idea of starting low, go slow, because the marijuana out there today, if you tried it in the 90s and now come back to it, now that it's legal, it's going to perhaps be a very different experience. And so this does go along with some of the data that we've seen from emergency room departments. It's interesting that Colorado is also noting an incidence of kind of vacation, people from out of town that are coming, like the tourism, marijuana tourism, where people are buying it on their vacation as a destination because it's legal, and then also underestimating the potency. And so we've got a lot of out-of-state individuals. So it'll be interesting to see if that happens in Oregon too. But there was a report from Colorado at the end of last year saying increased emergency room visits from out-of-state people who are purchasing marijuana, which really speaks to the fact that we need information at the point of sale. And it's certainly in Oregon dispensaries are mostly are owned and run by people that don't necessarily have any formal healthcare training. They often have a business background. So we know that we are one of the states where there isn't a requirement for any kind of messaging in that way. So just to mention, so the medicinal patients, this might not show up very well here, but they do have access to much, much higher serving sizes and higher concentrations. And so the retail market has been limited to avoid accidental exposure to vulnerable populations. But one reason that most medical patients will want to keep and have their card and go through that process is because they can buy much higher amounts. And so I'm not sure if this shows up very well here, but this is an example from a local dispensary, and it actually says, and you'll be able to see it on the slides, it'll give you the amount of THC and CBD in a serving size for the edibles. And the one at the top, it struck me because it's actually supposedly contains 362 milligrams of THC, which is actually higher than they're supposed to have in that brownie. But I believe there's more state visits these days in terms to regulate this. Just a few other terms that I guess keeping current is really difficult, especially in this field. And so hashish has always been known as a more concentrated form, or desired for its concentrated form of THC. This is coming from the resinous glands of, I believe, the female plant. But now we've got a big increase in interest in cannabis concentrates now that we've got them legal in Oregon. There's a new, so I'll show you here, this is from leafly.com because they can tell you how to do dabbing, and that's just where my information for this has come from. But dabbing is really, it sounds very dangerous. It's the flash vaporization of a cannabis concentrate with a solvent, and usually butane. You need a hot surface, and this is what the nail is. So this is the paraphernalia that you need for undergoing this new trend of dabbing. And then this very rapidly extracted concentrate is inhaled through a water pipe. And so the thing here is that although we've got those limits, I believe that this process, if I'm understanding it correctly, can produce very, very high concentrated, very quick hits of very concentrated cannabis. So I think these terms, butane, hash oil, shatter, wax, honeycomb, crumble, are all terms to describe what the concentrate looks like. So there's a multiplicity of new ways. I suppose it's bound to happen. Now it's legal. There's going to be new ways that people are using it, and increased dangers, I think. So just the heat of having the blowtorch, it sounds like a very hot, very quick, unpredictable kind of medical procedure. I sort of watch this space, but that's as much as I know about dabbing. And I guess it comes from these little tiny pots. That's where it comes from. So we've got it already concentrated forms, but then ways to make it more concentrated and really increase that dose. So in kind of thinking about this from both the recreational use of marijuana from the medical use, I found it was most easy for me to really think of what is the primary motivation, just to try and separate the medical population. And so really, I think there's a different goal. So symptom relief versus pleasure. And really, the people for which we approve the use of medical marijuana are those individuals that have severe debilitating medical conditions. So they already have a very high chronic burden of disease, and presumably they are feeling they need to reach and use medical marijuana because it's unmet need. So, but this population, I think we have to think of them as differently than the retail market. Now, there may not be a clear separation for some individuals, but the preferred route of administration may or might not differ either. So smoking is very rapid and predictable bioavailability. Self-titration, as to some extent, is the vapor. So again, we won't ever be able to see a human study where we can approve smoking marijuana. If we want to study this in human subjects, it's going to probably still be in a different form. And how well that actually equates to what's going on in terms of the population using it is you're still trying to extrapolate from the data that's available. So there's been a lot of messaging as well about orals, so that here, of course, the bioavailability is much more poorer. We're trying to educate people about activation time. So you take one and wait, don't just take another one. So dosing is still very difficult, whether it be medical or retail, the retail market. So if we look at the qualifying conditions in Oregon, and I will just say that these do differ from state to state, but Oregon lists cancer, glaucoma, and this is just the order that they appear in the Oregon statutes, degenerative or pervasive neurological conditions, HIV/AIDS, post-traumatic stress disorder, or we do allow it for that, although I would just mention at this point, I don't think really we've got good studies that really are going to show a clear benefit. medical condition with severe pain, severe nausea, seizures, including but not limited to epilepsy, and then persistent muscle spasms, which are included but not limited to those caused by multiple sclerosis. So we have a lot of conditions. Just to reiterate some of the numbers that you perhaps heard of earlier. So we've got about 67,000, this is as of January, just the statistics. So every quarter these are updated. We have over 67,000 cardholders. We've got a significant population of caregivers, and this also speaks to how functional are some of our medical marijuana patients, in that caregivers can hold the card to get and to obtain marijuana for the use of somebody else. We've got a lot of growers in the state, and we've got many thousands of grow sites all over the state, and producing slightly different sizes, and also presumably producing different strains. So we'll have local growers here, and again, a bit like the wine industry. We've got 1,722 physicians listed that have between 1 and 449 patients, and then a smaller number that have over 450 patients. That's just the way the Oregon Health Authority breaks it down. But really, just to speak to what is the primary, what is listed as the primary use, and severe pain is the majority of card holders cite that as their primary reason for holding the card. You can have more than one condition, but primary pain, and I've highlighted spasms as well, because it turns out we can tell a story of what's happening in other countries, and where we have the use of a pharmaceutical product that is approved for muscle spasms associated with multiple sclerosis. So if we think about marijuana for management of persistent chronic pain, neuropathic pain, cancer pain, and muscle spasms. And so I guess here we're thinking about pain that is not managed well by other means. And so there is good evidence. If we again look to the endogenous cannabinoid system, the endocannabinoids, they do seem to modulate pain perception under physiological conditions. There is some good evidence that the synthetic and endogenous cannabinoids are analgesic. And if you activate cannabinoid receptors in many animal models, so some of our preclinical data does show that there is some rationale for pursuing this in the pain pathway. So I think one way to think of it, and again we've really not tapped the lower receptor system for therapeutic advantage yet, but really we have two parallel endogenous pain pathways and we've actually used plant-derived natural products to learn more about these and then manipulate it for our own advantage. So we've got the endogenous opioid peptides, which act at specific receptors. And then really it appears we've got lipid modulators, the endogenous cannabinoids, that primarily with respect to pain are probably acting at CB1 receptors. So some of the signaling that is really where we can say, okay, well, where in this pathway may cannabinoids and therefore marijuana in kind of bulk exposure to cannabinoids or phytocannabinoids, how can this be working? So we do know that these receptors, because we know they're all over the body, they're in the brain, the spinal cord, and the periphery. It's clear that probably there is some important cannabinoid signaling in the dorsal horn of the spinal cord. And so we've got some clear and nociceptive responses. So this input here is regulated by, or modulated again by, inputs from neurons that are enhancing release of, here we've got neuroactive peptides. So this would be a pro-nociceptive response, things that releasing onto this synapse, and even glutamatergic input here. So having endogenous, the ability to inhibit that pro-nociceptive responses, that inhibitory effect, really gives us at least a working model and puts the receptor and the ligand in the right place to be real modulators of this response. We know that, oh, there you go. We know another area of intense interest is the active role of endocannabinoid receptors, so the CB1 receptor, in this descending pathway in terms of the perception of pain. So we know that in the brainstem, these lower brain regions, we've got in the periaqueductal gray region and the rostroventromedial medulla. So this classic pain descending pathway into here, we do have cannabinoid receptors that endogenously probably also have this disinhibition. So the role of the receptor is to inhibit ligand release, and we think that if there is a role in the perception of pain, it's because we know endogenously these systems are in place to naturally reduce our perception of pain. So there is some evidence that the receptors are there. This talk was made much easier by the publication of this report, which Dr. Hedberg also alluded to. So I quickly looked to see what now this report, and I've put the link here, the National Academy of Sciences, so a very large task force that was charged. We have been doing this on a state level, but now to look at a national level and come up with some better guidelines for the health effects of cannabis and cannabinoids. Really, pain emerges as one of the very few really indications where they will say that there is conclusive or substantial evidence that cannabis is effective for the treatment of chronic pain in adults. Oral cannabinoids are also effective treatments for improving patient-derived reported MS spasticity symptoms, although they conclude that there's limited evidence for an effective clinician-measured spasticity. So now we do have some clearer, in terms of looking at clinical data and clinical studies that are out there, this has been the conclusion of this this report. And so I think that at least gives us a milestone going forward. So the receptor and the signaling is there, and with the available data that we have for the control trials that have been good, pain does emerge as probably the indication that we should look to first in terms of finding a true an effect. I'll just mention cannabidiol because the idea of this an approach with two different, more than one cannabinoid present and having efficacy not with the combination of the cannabinoids. We know that cannabidiol, and this has been very famous at the moment, we're watching very carefully the results of some ongoing clinical trials where this has had orphan drug designation, so purified cannabidiol, so 99% as a purified extract to look at what kind of efficacy it has in children with intractable epilepsy syndrome. So those trials are ongoing. But we know that cannabidiol does not bind very well to the cannabinoid receptors. So there's been this question as where it does act. And now it seems that it probably binds intracellularly. So it probably enters the postsynaptic cell and actually this is really the emerging hypothesis that it inhibits the breakdown of the endogenous ligand. So although we were looking to advance small molecule inhibitors of termination of the endogenous ligand, it seems that we already have an endogenous degradation system, and it may turn out that this seems to be what the best guess at the moment is that cannabidiol actually has a completely different target. But by, again, inhibiting breakdown, you are going to have more functional levels of the receptor. So this is still a little bit confusing, but I think we shouldn't be looking at the plasma membrane as the potential target. Potentially there's intracellular binding sites. So just to look at what's going on in other countries, and that might give us some insight and where we can look and look to more data to see. So Sativex is another pharmaceutical product which is a standardized combination of THC and cannabidiol together. So now there's evidence that cannabidiol does not bind to receptors, but also is targeting the same pathway to enhance endocannabinoid transmission. And so this is 2.7 milligrams of THC, 2.5 milligrams of CBD. It's approved in Canada, Australia, several European countries for persistent muscle spasms and pain associated with multiple sclerosis. If it is approved, so at the moment it's in phase 3 clinical trials for unmet pain with respect to cancer. It may be, even if it's approved in Oregon, it might not be, we still don't know how much this would cost. But I think at the moment we're searching for answers with respect to medical marijuana and looking to these other pharmaceutical products that are out there and being approved by other countries might also help us give us some more options. And so if we have the availability of other formulations that are more standardized, that might help this field. And so this is called, if this is approved in America, this is actually Nabiximols, it's called, and Sativex is the trade name. It will become known as a non-opioid analgesic. So these are going to be new terms that may or may not appear for us. It's an endocannabinoid system modulator. And although we don't really have a clear idea of how it works in spasticity, this is what it's being used for. It's an oral, this is in French because this is the Canadian version, but it's an oral spray. So it allows mucosal absorption and self-titration, much as you would, as medical marijuana users are doing. So I think, we might be back to a tincture of cannabis again in terms of having this available. So, so just as I wrap up, so some of the condition, as a medicine, the psychoactive effects may not be well tolerated in naive users. If this was the only choice you had for a medicine, it might be very off-putting to actually just to navigate this whole network and this whole dispensary system, and the availability is very different. So if you're a naive user, may not be great. Definitive evidence for most therapeutic effects is still really lacking, and then we've got a lot of trouble associated with it being a non-pharmaceutical grade product in terms of quality control. So some of these sort of famous side effects, but just to have them in your notes in one place. So a very early onset, so we know that it increases heart rate, vasodilation, decreases, then kind of goes into a decrease of heart rate. Red eyes is a famous side effect. And this, we do have cannabinoid receptors in our eye. I should mention that it's not endorsed for glaucoma, though that is one of the listed uses in Oregon. So we know that it's going to impair short-term memory, complicates learning and ability to retain information, impairs motor coordination, driving skills, altered judgments and perceptions of safety. These are all really predicted through its effect as a central nervous system acting agent, and should largely be reversible if people abstain within 24 hours. Again, we're now starting to recognize adverse effects, so definitely frequency, this now is pretty clear. The frequency of use seems to be related to the use of other substances and the emergence of cannabis use disorder, negative effects on psychiatric illness, worsening of symptoms. And then now this is also a clear adverse effect, symptoms of bronchitis in those who are habitual smokers, so increased cough, sputum, and would be additive if the individual is also smoking cigarettes. This is an unusual, another counterintuitive side effect, but the emergence of cannabinoid hyperemesis syndrome, so this cyclical vomiting, and it seems to be associated with chronic or very heavy use, whether it be recreational or medical. And individuals really can only get relief through showering, apparently. So I put the shower head here. The symptoms stop after use of cannabis, but then will re-emerge once the person. So as related to cannabis use, heavy cannabis use, we've got this unusual effect. As probably people realize, this is a very lipophilic substance, so it can hang around in our bodies for a long time, making it very difficult to detect or to distinguish a habitual user from someone who's using it, just tries it once. So this is really interpreting the interpretation, the difficulties interpreting the immunoassays in terms of drug testing in the workplace. Drug-drug interactions, we know it should be contraindicated with other sedatives, just because we just don't know how exactly how. We don't have the data really, these would be combined effects. And then unfortunately in this whole area we're really relying on anecdotal reports of interactions or changes in the perception of how somebody felt, that they didn't enjoy the experience or it wouldn't work as well as they thought it would. And it's again because I feel that we're really in a phase of post-market evaluation because we already have the dispensaries and everybody's using it. But I guess you would say that it's relatively safe in monotherapy, and that's how we would conclude that. So in summary, there's very large gaps in our knowledge. I think the recent report, a review of the available data, I think this will turn out to be an area of much hope. The idea that there may be a potentiation or an alternative way to produce analgesia and reduce the dose of opioids, I think this is something that we need to work through. Again, just the medical marijuana users have unmet need in terms of their pain, presumably pain as well as other conditions. But although we've spent a long time studying THC, we don't really know the mechanistic basis of the endogenous cannabinoid system, and this will still limit us until we can get more research. So I'll just leave you with my last slide. You know, it may not always be this way. This is, I got this from a press release that coming from Israel, which has been at the forefront of utilizing marijuana. They have are able to produce pharmaceutical grade cannabis, which we don't do in Oregon. But this is their first metered selective dose vaporizer. There's the medical device that's going to be used, and so it'll be interesting to see if we're just living in the dark ages and eventually things will change, because certainly in other countries, we can look to other countries which are moving ahead with this and using it in hospitals. I'm sorry for going a little bit long, but thank you for inviting me.